Biochemistry, the chemistry of life. Some students are surprised to learn that they must study chemistry as part of a microbiology course. The reason why chemistry is an important component of a microbiology course is the answer to the question, what exactly is a microorganism? A microbe can be thought of as a bag of chemicals that interact with each other in a variety of ways. Even the bag itself is composed of chemicals. Everything a microorganism is and thus relates to chemistry. The various ways microorganisms function and survive in their environment depend on their chemical makeup. The same things are true about the cells that make up any living organisms, including human beings. The cells, too, can be thought of as a bag of chemicals. Biochemistry is the study of biology at the molecular level, and can thus be thought of as the chemistry of life or the chemistry of living organisms. Not only is biochemistry a branch of biology, but it is also a branch of organic chemistry. Biochemistry involves the study of the biomolecules that are present within living organisms. These biomolecules are usually large molecules called macromolecules and include carbohydrates, lipids, proteins, and nucleic acid. Other examples of biomolecules are vitamins, enzymes, hormones, and energy-carrying molecules such as adenosine triphosphate or the ATP. Humans obtain their nutrients from the foods they eat. The carbohydrates, fats, nucleic acids, and proteins contained in these foods are digested, and their components are absorbed into the blood and carried to every cell in the body. Within cells, these components are then broken down and rearranged. In this way, the compounds necessary for cell structure and function are synthesized. Microorganisms also absorb their essential nutrients into the cell by various means to be described later. These nutrients are then used in metabolic reactions as sources of energy and as building blocks for enzymes, structural, macromolecules, and genetic material. So now we were going to tackle the carbohydrates. What is carbohydrates? Carbohydrates are biomolecules composed of carbon, hydrogen, and oxygen in the ratio of 1:2:1, or simply CH2O. Glucose, fructose, sucrose, lactose, maltose, starch, cellulose, and glycogen are all examples of carbohydrates. The monosaccharides. The simplest carbohydrates are sugars, and the smallest sugars or simple sugars are called monosaccharides. From the Greek, mono meaning one, and the saccharon meaning sugar. The one refers to the number of rings. In other words, monosaccharides are sugar composed of only one ring. The most important monosaccharide in nature is the glucose or the C6H12O6, which may occur as a chain or an alpha or beta ring configurations. Monosaccharide may contain from 3 to 9 carbon atoms, although most of them contain 5 or 6. A 3-carbon monosaccharide is called a triose. One containing 4 carbon is called a tetrose, 5 a pentose, 6 a hexose, 7 a heptose, 8 an octose, and 9 a nonose. Ribose and deoxyribose are pentoses that are found in RNA and DNA, respectively. Glucose, also called dextrose, is a hexose. Octoses and noses are quite rare. The main source of energy for body cells, glucose, is found in the most sweet fruits and in blood. The glucose carried in the blood to the cells is oxidized to produce the energy-carrying molecule ATP, with its high-energy phosphate bonds. ATP molecules are the main source of the energy that is used to drive most metabolic reactions. Other monosaccharides are galactose and fructose, both of which are hexoses. The sweetest of the monosaccharides is found in fruits and honey. So next is the disaccharides. Disaccharides, di meaning two, are double-ring sugars that result from the combination of two monosaccharides. The synthesis of a disaccharide from two monosaccharides by removal of a water molecule is called a dehydration synthesis reaction. The bond holding the two monosaccharides together is called a glycosidic bond. It is a type of covalent bond. Glucose is the major constituent of disaccharides. Sucrose, or the table sugar, is a sweet disaccharide made by joining together a glucose molecule and a fructose molecule. Sucrose comes from sugar cane, sugar beets, and maple sugar. Lactose or milk sugar and malt sugar are also disaccharides. Lactase is made by joining together a molecule of glucose and a molecule of galactose. People who lack the digestion enzyme lactase needed to split lactose into its monosaccharide components are said to be lactose intolerant. Maltose is made by combining two molecules of glucose. Disaccharides react with water in a process called a hydrolysis reaction, which causes them to break down into two monosaccharides. Peptoglycan is a complex macromolecular network found in the cell walls of all members of the domain bacteria. Peptidoglycan consists of a repeating disaccharide attached by polypeptides or proteins to form a lattice that surrounds and protects the entire bacterial cell. A number of antibiotics, including penicillin, prevent the final cross-linking in the rows of disaccharides, thus weakening the cell wall and leading to lysis or bursting of the bacterial cell. Although most members of the domain archaea have cell walls, their cell walls do not contain peptidoglycan. The polysaccharides. The definition of a polysaccharide varies from reference book to another, with some stating that a polysaccharide consists of more than 6 monosaccharides, others stating more than 8, and others stating more than 10. Means many, and in reality, most polysaccharides contain many monosaccharides, up to hundreds or even thousands of monosaccharides. Thus, polysaccharides are defined as carbohydrate polymers containing many monosaccharides. Examples, including starch and glycogen, which are composed of hundreds of repetitive glucose units held together by different types of covalent bonds, known as glycosidic bonds for glycosidic linkages. Glucose is the major constituent of polysaccharides. Polysaccharides are examples of polymers, molecules consisting of many similar subunits. Some of these molecules are so large that they are so insoluble in water. In the presence of the proper enzymes or acids, polysaccharides may be hydrolyzed or broken down into disaccharides and then finally into monosaccharides. Polysaccharides serve two main functions. One is to store energy that can be used when the external food supply is low. The common storage molecule in animals is glycogen, which is found in the liver and in muscles. In plants, glucose is stored as starch and is found in potatoes and other vegetables and seeds. Some algae store starch, whereas bacteria contain glycogen granules as a reserve nutrient supply. The other function of polysaccharides is to provide a tough molecule for structural support and protection. Many bacteria produce polysaccharide cocktails, which protect the bacteria from being phagocytized or eaten by white blood cells. Cellulose is another example of a polysaccharide. Plant and algal cells have cellulose cell walls to provide support and shape as well as protection against the environment. Cellulose is insoluble in water and ingestible for humans and most animals. Some protozoa, fungi, and bacteria have enzymes that will break the B-glycosidic bonds linking the glucose units in cellulose. Some of these microorganisms, saprophytes, are able to disintegrate dead plants in the soil, and other parasites live in the digestive organs of herbivores or the plant eaters. The lipids. Lipids constitute an important class of biomolecules. Most lipids are insoluble in water but soluble in fat solvents, such as either chloroform and benzene. Lipids are essential constituents of almost all living cells. The fatty acids. Fatty acids can be thought of as the building blocks of lipids. Fatty acids are long-chain carboxylic acids that are insoluble in water. Saturated fatty acids contain only one single bond between the carbon atoms. Fats containing saturated fatty acids are usually solids at room temperature. Monounsaturated fatty acids, such as those found in butter, olives, and peanuts, have one double bond in the carbon chain. Polyunsaturated fatty acids, such as those found in soybeans, sunflowers, and corn, contain two or more double bonds. Most fats containing unsaturated fatty acids are liquid at room temperature. The terms saturated, monounsaturated, and polyunsaturated fatty acids are often heard in discussions about human diet. Certain fatty acids called essential fatty acids cannot be synthesized in the human body and thus must be provided in the diet. For purposes of discussion, lipids can be classified into the following categories: the waxes, fats and oil, phospholipids, and glycolipids, which will be discussed later. Waxes. A wax consists of a saturated fatty acid and a long-chain alcohol. Wax coatings on the fruits, leaves, and stems of plants help to prevent loss of water and damage from pests. Water on the skin, fur, and feathers of animals and birds provide a waterproof coating. Lanolin, a mixture of waxes obtained from wool, is used to hand and body lotions to aid in retention of water, thus softening the skin. The waxes that are present in the cell walls of Mycobacterium tuberculosis, the causative agent of tuberculosis, are responsible for several interesting characteristics of this bacterium. For example, should A. M. tuberculosis cell be phagocytized by a phagocytic white blood cell, a phagocyte, the waxes protect the cell wall from being digested. This enables the bacterial cell to survive and multiply within the phagocyte. Also, the waxes in the cell walls of M. tuberculosis make the organism difficult to stain, and once stained, the waxes make it difficult to remove the stain from the cell. The fats and oils. Fats and oils are the most common types of lipids. Fats and oils are also known as triglycerides because they are composed of glycerol, a three-carbon alcohol, and three fatty acids. Fats are triglycerides that are solid at room temperature. Most fats come from animal sources. Examples include the fats found in meat, whole milk, butter, and cheese. Most oils are triglycerides that are liquid at room temperature. The most commonly used oils come from plant sources. Olive oil and peanut oil are monounsaturated oils, whereas oils from corn, cottonseed, safflower, and sunflower are polyunsaturated. The phospholipids. Phospholipids contain glycerol, or the fatty acids, a phosphate group, and an alcohol. There are two types: glycerophospholipids, also called phosphoglycerides, and sphingolipids. The glycerophospholipids are the most abundant lipids in cell membranes. The basic structure of a cell membrane is a lipid bilayer, consisting of two rows of phospholipids, arranged tail to tail. The hydrophobic tails, lacking an affinity for water molecules, point toward each other, enabling them to get as far away from water as possible. The hydrophilic heads, being able to associate with water molecules, project to the inner and outer surface of the membrane. Two other types of lipids are also found in eukaryotic cell membrane steroids, primarily cholesterol in animal cells, and glycolipids. The cell membrane also contains proteins which have been described as iceberg floating in sea of lipids. In addition to phospholipids, the outer membrane of gram-negative bacterial cell walls contain lipoproteins and lipopolysaccharide, or the LPS. As the name implies, LPAs consist of lipid portion and a polysaccharide portion. The lipid portion is called lipid A or endotoxin. When endotoxin is present in the human bloodstream, it can cause very serious physiologic conditions, example fever and septic shock. The cell walls of gram-positive bacteria do not contain LPS. The glycolipids. Glycolipids are abundant in the brain and in the myelin sheaths of nerves. Some glycolipids contain glycerol plus two fatty acids and a monosaccharide. Cerebrosides and gangliosides are examples of glycolipids. Both are found in the human nervous system. A person's blood group, AB, A, B, or O, is determined by the particular glycolipids that are present in the surface of that person's red blood cells. The proteins. Proteins are among the most essential chemicals in all living cells, referred to by some scientists as the substance of life. Some proteins are the structural components of membranes, cells, and tissues, whereas others are enzymes and hormones that chemically control the metabolic balance within both the cell and the entire organism. All proteins are polymers of amino acids. However, they vary widely in the number of amino acids present and in the sequence of amino acids, as well as their size, configuration, and functions. Proteins contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. The amino acid structure. A total of 23 different amino acids have been found in proteins. 20 primary or natural occurring amino acids plus 3 secondary amino acids derived from primary amino acids. Each amino acid is composed of carbon, hydrogen, oxygen, and nitrogen. 3 of the most have sulfur atoms in the molecule. Humans can synthesize certain amino acids but not others. Those that cannot be synthesized, called essential amino acids, must be ingested as part of our diet. The term essential amino acids is somewhat misleading. However, in view of the fact that all of the amino acids are necessary for protein synthesis, because we cannot manufacture the essential amino acids, it is essential that they be included in our diets. The protein structure. When water is removed by dehydration synthesis, amino acids become linked together by a covalent bond referred to as peptide bond. A dipeptide is formed by bonding 2 amino acids, whereas the bonding of 3 amino acids forms a tripeptide. A chain polymer consisting of more than 3 amino acids is referred to as a polypeptide. Polypeptide is said to have primary protein structure, a linear sequence of amino acids in a chain. Most polypeptide chains naturally twist into helices or sheets as a result of the charged side chains protruding from the carbon-nitrogen backbone of the molecule. This helical or sheet-like configuration is referred to as secondary protein structure and is found in fibrous proteins. Fibrous proteins are long, thread-like molecules that are insoluble in water. They make up keratin, found in hair, nails, wool, horns, feathers, collagen in tendons, myosin in muscles, and in the microtubules and microfilaments of cells. The enzymes. Enzymes are protein molecules produced by living cells as instructed by genes in the chromosomes. Enzymes are referred to as biological catalysts, biologic molecules that catalyze metabolic reaction. A catalyst is defined as an agent that speeds up a chemical reaction without being consumed in the process. In some cases, a particular metabolic reaction will not occur at all in the absence of an enzyme catalyst. Almost every reaction in the cell requires the presence of a specific enzyme, although enzymes influence the direction of the reaction and increase its rate of reaction. They do not provide the energy needed to activate the reaction. Some protein molecules function as enzymes all by themselves. Other proteins called apoenzymes can only function as enzymes. For example, can only catalyze a chemical reaction. After being linked up with a non-protein cofactor, some apoenzymes require metal ions, for example, the Ca2, Fe2, Mg2, copper2, or referred as the manganese and copper, as cofactors, whereas others require vitamin-type compounds called coenzymes, such as vitamin C, flavin adenine dinucleotide or the FAD, anicotinamide adenine dinucleotide NAD. The combination of the apoenzyme plus the cofactor is called haloenzyme, a whole enzyme. The whole enzyme can function as an enzyme. Apoenzyme plus cofactor equals haloenzyme, a functional enzyme. Enzymes are usually named by adding the ending "ase" to the word, indicating the compound or types of compounds on which an enzyme acts or exerts its effect. For example, proteases, carbohydrates, and lipases are enzymes that exert their effects on proteins, carbohydrates, and lipids, respectively. The specific molecule in which an enzyme acts is referred to as that enzyme's substrate. Each enzyme has a particular substrate on which it exerts its effect. Thus, enzymes are said to be very specific. Although most enzymes end in ase, some do not. Lysosome and hemolysins are examples.